Okay, so we're going to have our next presentation from Abira Rajah. So she's also a former student from my lab who did her honors and master's project with me. And she's a research scientist in the biotechnology industry specializing in cancer drug discovery. She has her master's of science in physiology from McGill. And as I mentioned, did her thesis research with me. She's contributed to the target discovery platform at Repair Therapeutics and is currently advancing similar efforts at GBIxBIna, focusing on uncovering novel tumor vulnerabilities for therapeutic intervention. So thank you for being with us today.
So hi everyone, I'm happy to be here today. So as Claire mentioned, I'm currently a research scientist at a biotech startup based in Montreal called GBIxBIna. So I joined Dr. Claire Brown's lab in 2017 for a project funded by NSERC, which then extended to a full year's honors project as part of my undergraduate program, and then extended to a 2 years master's research project. So from 2017 to 2020, Dr. Brown and I were able to publish 2 papers that explored how the focal adhesion protein paxillin can regulate cell migration and directionality. So cell migration is a process that should be studied at multiple different scales. So traditional wide field or confocal microscopy can be used to understand cell migration at a population level, but more sophisticated modalities such as turf microscopy can be used to study the single cell as well as subcellular structures inside the cell during migration. So in this paper, we found that during cell migration, adhesions at the protrusive and retractive edges have different rates of the protein paxillin binding at the adhesions, and we also found that it was regulated by the phosphorylation state of paxillin. So all the images that we acquired were at the ABIF, and all the analysis was also done at the ABIF. And the paxillin binding rates, that analysis was actually done through a technique called temporal image cross-correlation microscopy. So the ABIF was actually able to bring in-house at this analysis from a group at the University of California in Irvine in order to help advance our research. So what I realized from my experience is that having access to a core facility such as the ABIF is not only about access to the instruments and the image analysis softwares and the imaging experts, but also having access to workshops and seminars meant to help researchers get practical exposure to newly emerging technologies. I had the privilege of participating at workshops organized by the ABIF, such as the Montreal Light Microscopy course in 2018, as well as the Super Resolution Microscopy course back in 2019, organized by the Neuro. And thanks to these courses, I was able to get the foundational knowledge as well as hands-on training of not only traditional microscopy techniques, but also advanced techniques such as STED, Palm, Storm, directly from vendors who created these tools. So the microscope was an indispensable tool for my research projects at McGill University, but I have not stopped imaging since I graduated from my master's. In fact, I understood the impact the ABIF and Dr. Brown had on the scientific community in Montreal is when I joined as a research scientist at my former company, Repair Therapeutics, and my colleagues were just excited because they knew what I was bringing in terms of microscopy knowledge. So in the R&D world and in the pharmaceutical world, imaging experiments are actually constantly done to develop life-saving medications. And it can be to study, for example, drug dose response in real time. So we could measure cell viability, protein phosphorylation states, cell cycle states in real time in response or to a therapeutic. We could use imaging to visualize drug distribution in vivo in animal models. And in the clinic, we can also use it to identify new biomarkers directly in patient tissues. So listed here are only some applications used probably every day to discover new drugs. And I just want to mention that research institutions or companies like us requiring these kinds of experiments for the drug discovery projects can benefit from the presence of core facilities such as the ABIF in their local scientific ecosystem, as they can support setting up these kinds of assays or allowing access to the equipments that enable these experiments. I think we can all agree that the microscope is an evolving technology with new modalities and applications emerging frequently to advance biological research. Core facilities such as the ABIF play a role in accelerating these kinds of discoveries. And as an alumnus, I think the support and knowledge presented by the ABIF and Dr. Brown had an immeasurable impact on my career, as well as many other researchers in McGill and in Canada. So thank you for all you do. That's it.
Thank you, Abira. So we have time if there's any questions. I know you mentioned that you brought a lot of expertise into the company.
Yeah.
What kind of imaging expertise did they already have? Was it relatively routine, or?
It was like we already had several microscopes, but a lot of the focus was on high content screening. But like if there was new applications, for example, like immunofluorescence techniques and live imaging, I think it was nice to bring in that kind of expertise. Yeah.
And do you think the expertise you had played a role in securing the position?
I would say for sure. A lot of my, when I was applying for the jobs, like you do have to give a seminar on your research projects, and most of my project was imaging. So I think that already says that how imaging was important for them, having that skill set to get the job.
Okay. Any other questions? Yes.
Thank you very much for your presentation. Maybe to bounce back on this question, I was wondering if, like, with all of the expertise that you brought with you, were they like asking you to maybe have like a decisive role into acquiring new equipment or like to design new experiments, especially because I think you mentioned it was a startup? And also, maybe could you develop a bit more about if you had like specific skills that you developed during your academic experience at the ABF that you leverage during your now industry jobs, whether it's like hard skills or like more soft skills that you developed during this experience?
I think all the skills that I developed during my academic career was super important, and I apply it every day in industry. Like we are doing the same science that we do in academia versus in industry, but it's at a faster pace. So we're not always highly focused on getting to the nitty-gritty detail of a biological phenomenon, because we are in the mission of discovering new drugs, so we need to find the fastest path forward. But all the types of skills, like wet lab skills, communication skills, presentation skills, it directly transfers into what you need in industry.
So you can join me again in thanking Abira.
Thank you.